Good afternoon, everyone. Thanks for the opportunity to present today. I'm going to be presenting Oxford Biomedica, where I am leading the commercial efforts, including the sales team, marketing, and strategy, as well as corporate development. We have positioned ourselves as a quality and innovation-led CDMO specialized in viral and gene therapy. And it's really part of our strategy. A number of CDMOs have decided that they wanted to work in multiple modalities. We've made the opposite decision. We want to stay focused on only one thing, what we do best, manufacturing viral vectors. And I will drive you through a number of key facts and data about the company in the next 10-ish minutes. As we're a listed company, let me start with some looking statements that I'm going to pass quickly and introduce the company. So we've been operating in that space for more than 25 years. The company was actually created in 1995. And we are operating from the very beginning of the value chain with the gene of interest, the design of the vector, the optimization of the vector, through process and analytical development, clinical manufacturing, and finally commercial manufacturing as well. We are today running a process for a vector that is a commercial vector and approved in more than 40 countries. Last time I checked the number, it was 43 countries, to be extremely accurate here. And although we've been known during many years as a company who was working on lentiviruses, we work on multiple viral vectors. We do deliver lentiviruses, but also AAVs, adenoviruses, but also VSV, HSV, and a number of other vectors. The list is quite long. Looking at the past 10 years, where the vast majority of the activity happened, we've delivered shy of 400 successful GMP batches. And looking at the number of projects on which we work today, we're working on more than 40 active programs in parallel at different stages, obviously, process development, clinical manufacturing, or commercial manufacturing. If we're looking at the CGT space only, we have one successful BLA submission. If we're looking at CGT and vaccine manufacturing, since we manufacture the vaccine for AstraZeneca, then we have two successful BLA submissions. And we have more than 30 successful IND submissions, again, looking at the past 10 years. We're also known as a company that is operating primarily based on its technology platform. Well, it's true and it's not true. Indeed, we have developed our platforms on lentiviruses and AAVs, but we're also taking a number of projects where our clients are coming to us with a vector and they want either optimization or directly process development or directly manufacturing. We have a number of sites. We usually say we have 2 locations, which is true, but we have more sites than 2. We have 4 sites out of Oxford, UK, and 1 site in Bedford, Massachusetts, USA. In Oxford, you see 4 sites here on the slide. The first one being our analytical development and process development center, Windrush Cart, where we really have labs that are amazing. I remember the first time when I visited, what struck me was the level of automation. And indeed, we know that what we do is extremely labor intensive, so investing in automation has been a key theme in the company to make sure that we were increasing the throughput, and I'll come back to that in one of the next slides when we touch innovation. We have 3 bioprocessing centers, as you see here on the slide. Most of them are FDA and MHRA approved, and when I say MHRA, I should actually say MHRA and EMA approved. The biggest one being called Oxbox, where we have 4 GMP suites, and we have a plan to expand and bring online another 2 GMP suites as we're increasing our commercial manufacturing footprint with the demand coming from our clients, and programs that have been successful in phase 1, then phase 2, and are today in phase 3 at process characterization or process validation stage. The site in Bedford is a site that we acquired about 18 months ago now, where the technology that had been developed there was on AAV. As of now, we're tech transferring lentiviruses in US and AAV into UK so that we can deliver all platforms from all the geographies. So more to come on that, and I have a slide later to cover the future expansions. Talking about innovation, many people look at innovation like breakthrough innovation. That's not the way we look at innovation. We're looking at innovation as any kind of activity that helps moving the needle. So it can be maximizing data integration and analysis, working with artificial intelligence, working with algorithm to make faster decisions, to improve the sequence of our vectors. It can be automation that I mentioned before. It can be around the vector themselves to target these vectors or optimize the level of expression, minimize the RNA's transcripts that you don't want, and maximize the RNA transcript that you do want. It can be about improving the existing generation of AAV or LAN-T vectors that we have in-house to make sure that we increase the productivity, but not only, that we increase the quality level of the products coming out of a standard GMP batch. We're working on optimizing the productivity not only through the design of the vector, but through the design of the process. So we have a standard process that we're using, suspension. We've optimized that process to run it in suspension and perfusion so that we can push up the productivity. And the average increase in productivity when we move to perfusion is about 4 times more than the suspension non-perfusion process. Last but not least, we're working on producer cell lines or packaging cell lines. Not all the clients have the same needs, so we're trying to innovate through the entire portfolio of tools that we're proposing as part of our toolbox that we usually call the platform. The recent innovation that we have delivered, I just mentioned the suspension process, so suspension perfusion, so I'm not going to come back on that. Just one point, increasing productivity, but I also mentioned it, increasing the quality as well by increasing the purity of the vector that is produced. We've launched our 4th generation of LAN-T virus vectors that has an improved quality but also an increased potency and a higher capacity. We can increase the size of a gene of interest by 1 kb in TetraVecta, that is the brand name we're using for that platform, versus the 3rd generation. We've delivered recently high-titer packaging cell lines as well. We've scaled up AAV from 500 liter to 2,000 liter. And we've also automated, I mentioned automation as part of the innovation, we've automated the RCL assay, which we know is part of the bottlenecks that the industry is facing right now. I said it before, we're delivering end-to-end. That's true for any platform that we manipulate. That's true for LAN-T, for AAV, but also for adenoviruses. And when we say we deliver end-to-end services, we don't forget that there are key aspects of the projects that we run. It's not only about technology, it's also about making sure that the QA release can be done on time and quickly, because we know that we need that material for patients. It's also about being supportive at every step of the project, whether it's on the strategy very early or at the very end on the CMC support. So that's what we mean by end-to-end services. We do have plans to expand our capacity. It's been made public in the news about 2 weeks ago. We're currently buying 2 sites in France because we needed additional footprint in the process and analytical development space, but we also wanted to be closer from our clients in continental Europe. So we're adding 2 sites who have experience and expertise in vaccine manufacturing in the cell and gene therapy space. Like Oxford Biomedica, they've been operating since 1995, so quite a significant experience here. They're bringing to our larger organization multiple platform experience, including measles, MVA, vaccinia, I mean, you name it, the list is long here. Suspension and adherent platforms here, and they've been operating as a CDMO, so for us it was a natural way to expand our footprint and bring more capabilities and capacity to our clients who actively demand for more. So the new Oxford Biomedica network will have expanded capacity and capabilities. And we want to make sure that we continue the investment, especially at process characterization stage, where we see a lot of demand these days, especially to have access to slots quickly. Time is of the essence, as we know in our industry. But it's also bringing to us complementary capabilities with, I believe, one of the most compelling offering in the viral vector space. And we're eager to welcome new colleagues and see how much we can cross-fertilize between the existing sites. I wanted to make sure I was saving time for question, and I think we have about 4 minutes if there are any questions in the audience. Thank you. No, if no question, you can meet us during the rest of the event anywhere. So wish you a good week. Thank you.